All right, we are here to do the replication follow along notes for DNA replication. A little bit of review. DNA replication is a semi-conservative process, which means that the parent template strands are then copied with a new daughter strand. And so it's parent-daughter and then parent-daughter are the two new DNA chromosomes that you get. Please remember that we form replication forks to copy DNA. Eukaryotic chromosomes can have hundreds of thousands of replication forks happening at any moment, and the purpose of that is to replicate the DNA as quickly as possible. The purpose of today is to get to know the players, each individual enzyme, and the job that they do in replication. So I'm going to share my camera, my dot camera, and this is the follow along notes DNA replication that I sent to you in your packet and or I hand it to you if you are an in-class student. It was 3 separate pages. I have cut them out along the dotted lines and I have taped them on the back to hold them together. And I try to tape them so that they're lined up as perfectly as possible. There is, of course, some error there because I am only human. So now is the time, if you need to do that, if you need to get your papers together, to go ahead and cut it out and tape it or staple it or do something to hold them together so that you can follow along. If you don't have those capabilities currently at home, that's perfectly all right. You will simply do each individual paper. You can have them side to side to side and then work on it that way. But don't lose one when you are studying for your exam. Okay, so the enzymes that we are going to talk about today, I realized I don't have a pen, sorry. The enzymes we are going to talk about today are, of course, DNA helicase, topoisomerase. Where's number 3? Number 3, single-stranded binding proteins, RNA primase, DNA polymerase 3, nucleoside triphosphates. We're going to talk about the differences in the leading strand and the lagging strand. You have DNA polymerase 1, sorry, I stuttered there, DNA ligase, and then exonucleases. And so that's really only the new term for you on this sheet. This is just so you have a giant overview of DNA replication in front of you to study and possibly to take your test with. I'm not a huge fan of people having notes out on their tests, but once again, what can I do about it? The first thing we like to do in our follow along notes is to color each individual thing as we talk about it. So helicase, you don't have to make my colors your colors. I'm gonna do this limey green color for helicase, and I'm just gonna, color it in, outline it, do something. I think I'm gonna just make a swirl like that apparently. And I'm gonna find my DNA helicase on my example here, and I'm also going to color it in. This is an art class. I'm not looking at your examples. Your coloring doesn't need to be perfect. I'll go ahead and do that here too. Okay. So, DNA helicase. Who is he? He, well, or she, it's an enzyme, it doesn't have a gender, is looking for a particular region in the DNA to bind to. So DNA helicase, do I need this? Nah. Is looking for an adenine thymine rich region to bind to. So it scans along the DNA, and when it finds this AT rich region, remember I told you all about the TATA box region for primase in your replication video, it's kind of the same deal. It's looking for that AT rich region and it binds. Once it binds, its job, as you well know, is that it unwinds DNA by breaking hydrogen bonds at the replication fork. So it looks for that AT-rich region to bind. And then once it's there, it starts breaking those hydrogen bonds. Now, where are the hydrogen bonds in DNA? If you said between the base pairs in the center of the helix, you are correct. So it is breaking the hydrogen bonds that we find in between. So like here, it is breaking the hydrogen bonds that we find in between these base pairs so that the template can unwind. Any place that helicase is the very beginning of our replication fork, because it has to break a bond or a hydrogen bond for that fork to start. Okay, good. As helicase unwinds DNA, we need to bring in topoisomerase. So I always think, I don't know why, but I always think of topoisomerase as blue. I don't know why, just what my head does. Seems like a blue guy. So let's find our number 2 here. Sorry, that's Mr. Topoisomerase. Now it shows topoisomerase being very close to helicase in this picture. That's not necessarily the case. He's probably a few hundred to 1,000 base pairs down the DNA strand. What is topoisomerase's job? He goes farther down the DNA strand, and he or she, once again, I don't know why I'm doing that, relieves twisting tension. Recall, if you will, my little rubber band example. Apparently I threw him somewhere. Okay, you have a rubber band, right? And you twist 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 and you keep twisting, and there's a lot of twisting happening. Please excuse me while I twist. Okay. And so here is our DNA all twisted up as a helix, right? So what's happening here is that we're starting a replication bubble somewhere in the middle of this DNA. Apparently I twisted too much. Oh goodness, pull apart. It's hard to do without somebody else doing it. So we're starting a replication bubble in the middle of that DNA helix, and you can see as I pull the bubble apart, down here, the rubber band gets tighter and tighter and tighter. This is exactly what would happen. This is exactly what would happen in our DNA as well. That can't happen because eventually it's going to get so tight that you can't unwind the DNA any farther, which stops replication synthesis or DNA replication, which stops the whole cell cycle, and then your cell has to die. So topoisomerase comes on down the strand and relieves a lot of this tension so that it can go back to being usable DNA. And it does that, and then it breaks off, and then it goes down farther, and it does it, and it breaks off, and it goes down farther, and it does it. And so that's how topoisomerase works. That's the purpose of it there. How does it relieve the twisting tension? It cuts the covalent phosphodiester bond, ahead of DNA helicase. So DNA can untwist. It's important that you know that helicase is breaking the hydrogen bonds. Topoisomerase is breaking the covalent bonds. You can put phosphodiester. I mean, you need to write that down, but covalent versus hydrogen is what I really need to know. Finally, It's not really important to our purposes, but just in case you ever come across it in a textbook or in your future, topoisomerase is also known as DNA gyrase. It has two names, actually. That's an E right here, sorry. DNA gyrase. Okay, so we have helicase. It has found that AT-rich region. It has bound. It is unwinding DNA, creating that replication fork. We have topoisomerase relieving the extra tension down the DNA strand. Now that we have broken the hydrogen bonds, we need to do something to make sure that they don't automatically form back together. What is it that we add there so that the bonds do not automatically form back together? If you're looking at number 3 and you're saying these words, then you would be correct. We add those single-stranded binding proteins. This is the color, I don't know, it's like some sort of peachy color I'm using. Obviously, you don't have to use my colors. Just make sure you are following your own color scheme. We have these single-stranded binding proteins here. The purpose of these, and they're going to be on either strand at either end of the replication fork, by the way. It doesn't show it over here, but at the other replication fork that's at the end of this bubble, there's single-stranded binding proteins. They are shortened. They are typically abbreviated as SSBPs. Single-stranded binding proteins. And the whole purpose of them is to stop re-annealing. I've said the word many times. Here it is in writing. To re-anneal means to form the hydrogen bonds back immediately after they're broken and to just stay your template strand. That would not be helpful. So they stop re-annealing at the replication fork. DNA wants to rebond because that's stable. So DNA wants to be stable, but we can't let that happen. So the SSBPs stop the parent strands from rejoining. And that is their purpose, which is good and helpful because then we can have room for our primase and our DNA polymerases and our ligase to come in and do their job. So SSBPs are essential. They're essential workers. Isn't that nice? Okay. We've talked about in the replication video DNA polymerase 3 and how it comes in and starts adding nucleotides to the 3' free end of something. But DNA polymerase 3 doesn't just create DNA out of thin air. It has to have something in place, a short little sequence in place to bind to start adding nucleotides. And that short little sequence is made out of RNA. And so we have to have RNA primase come in and create an RNA primer. So I'm going to use pink, yeah. It's like a pinkish purple. And we have RNA primase. Yep. And there's actually a few of them, of course, in this picture because we have to form primers everywhere that we want DNA polymerase to bind. So you have one on the leading strand here, the one that's building 5' to 3'. And I'm going to go ahead and just color the primer as well with the same color as the primase, even though it's already shaded differently, just to point that out. And then we have another primer here, starting the primer for a next lagging strand, an Okazaki fragment. So I'm going to color him as well. And then remember, our lagging strand has to be built in bits. So it's multiple primers to build multiple little strands. So I'm going to just go ahead and highlight all these primers with the purple pink that I have in my hand. Be careful not to color over other enzymes that are working here. Okay, good. So now that I have my primase and primers colored in, I'm going to go back to my little RNA primase square. And we're going to tell you what RNA primase does. I already told you verbally multiple times, but we're going to write it down. RNA primase builds short, complementary strands. We call these primers, also known as primers, of RNA. No, RNA, sorry, sorry, RNA. Don't do what I did, RNA. We have to have this. This tells DNA polymerase 3, and I'm going to shorten that to poly, DNA polymerase 3, where to start. Or it's a jumping off point for DNA polymerase 3, whatever. DNA polymerase 3 can't just start creating DNA nucleotides out of anywhere. It has to have a 3' in to bind something on to. And so we have to have the primers with a free 3' end so that DNA polymerase 3 can start adding nucleotides. Okay. Okay. I'm gonna run out of colors. Let's see. Okay, so we've mentioned him, the man, the myth, the legend, multiple times, but now we have DNA polymerase 3 to talk about. Here he is, a little sack-like guy. I'm gonna color him green. Green means go. I feel like going is a good color for him. So I have so he's number 5. So I have a number 5 here. So here's my DNA polymerase 3. And then here is another DNA polymerase 3 down here. He's going to bind to the lagging strand. So I'm going to, or bind so he can start building a lagging strand, sorry. And then here is one that is already connected to a lagging strand, and it's doing its job, or the primer of a lagging strand, once again, not the correct words. And that's it. Those are our DNA polymerase that are in play in our picture. You got 3 of them, 1, 2, 3, plus the example one. All right, DNA polymerase 3, as you know, its job, it was the 3rd DNA polymerase found. Let's say that. That's why it's number 3. DNA polymerase 1 was found first, and we'll talk about him in a minute. He's down here. And then you have DNA polymerase 2, which happens to be prokaryotic. And that was found second. And then we found the eukaryotic one. DNA polymerase 3 builds DNA in a 3' to 5' template. Sorry, on a 3'5" to prime time template. Let me say it that way. So daughter strand is 5' to 3'. It does this by adding nucleotides starting at the free 3' end of the RNA primers. Okay, oops, sorry, there we go. So it builds DNA on a 3' to 5' template. So the daughter strand is 5 to 3. So it builds 5 to 3, right? It adds nucleotides starting at the free 3' end. It always has to be a free 3' end. It cannot be anything else. But it has to start on those RNA primers because once again, it can't just build DNA off of nothing. It has to start somewhere. I'm going to get started on kind of color coding all my nucleotides or my nucleoside triphosphates, if you want to call them the fancy word, while you'll get that written down. I'm going with a bright coral color, just highlighting the nucleoside triphosphates that are coming in. Apparently I covered one up. And we're just gonna, yep, there's a lot of them. You don't really have to take your time to do this, but since I'm showing you everything on here, I thought we might as well highlight them. All right. Good, that should have been enough time for you to write everything down. And I'm going to come over here to number 6, our nucleoside triphosphates, AKA our nucleotides. The nitrogenous base with the ribose sugar that comes in as a triphosphate, but then we take away 2 phosphates, use that energy to bind it covalently as a monophosphate. Okay. So the triphosphates. These are your nucleotides. Okay, so I'm gonna write that down. These are your nucleotides, except they have triphosphates. This is the triphosphates. Okay. What we do that you saw in the replication PowerPoint and that you'll see in the videos is that we use the energy from removing, wow, I just totally messed up spelling moving. Removing 2 phosphates to form a phospho. I'm gonna go ahead and diester bond to create DNA. So the triphosphates come in, right? DNA polymerase is here. The triphosphates come in. And what DNA polymerase is really facilitating is the removal of 2 of the phosphates and utilizing that energy to covalently bond your nucleotides in place. Remember, you have ATP, GTP, TTP, and CTP. So you have a triphosphate for every one of the nucleotides that we use: adenine, guanine, thymine, and cytosine. It's a true story. Giving you a second to write that down. All right, as we know, one daughter strand is going to be lucky and be built 5 to 3' continuously, and we call this the leading strand. So let's talk about that leading strand. It only needs one, oh my God, I don't know what's wrong with me, one DNA polymerase. I know how to spell, I'm just not doing it well. DNA polymerase. We only need one DNA polymerase on that leading strand. It is built in the same direction as the replication fork. It is built continuously by adding nucleotides to the free 3' carbon. And that is how the leading strand do. In the interest of my time and my voice, you can pause me anytime you need to continue writing, but I'm going to go ahead and then we're going to talk about the lagging strand. Okay, these are also known as Okazaki fragments, after the guy who discovered their creation. And what the lagging strand has to do, it is being built 3' to 5', right? You know this by now. We can't build 3' to 5'. So we will have to come in and build very short sequences, 5 to 3, and then merge those all together with DNA ligase so that we can create a continuous strand. So they're also known as Okazaki fragments. It is built in discontinuous fragments, as we know. Needs multiple DNA polymerase 3s. And it is built in the opposite direction of the replication fork. I would very much like to point out that you start a replication bubble, right? You add a polymerase 3 and it starts going. But you continuously continue to expand that replication bubble. So what actually ends up happening is even the leading strand is going to need DNA ligase to come in and bind together the next polymerase 3 that's going to start over here and build it this way. And then the replication bubble is going to get bigger and another polymerase 3 is going to bind, right? Eventually, you get so big, the replication bubble, that both sides of the bubble are going to have a leading strand, and then both sides are going to have Okazaki fragments. So never does one side just get built perfectly without any kind of fragments being created. Okay, so then, sorry. We've got to bind things. No. Then we have our proofreader. Proofreader and also our primer remover. We have DNA polymerase 3. For lack of a better color, I'm going to make it brown. Making it brown. I said 3. I misspoke because that's just who I am right now. I, of course, mean DNA polymerase 1 because that's who we are talking about. DNA polymerase 1 is our proofreader and our primer remover. Okay, so there's our DNA polymerase 1. Right, and it does the two things I just talked about. We know that DNA polymerase 3, you know this from the replication lecture, 1,000 base pairs per second, but because it's so fast, it has a high error rate, right? DNA polymerase 1 is 20 base pairs per second. Okay, so it's much slower, and because it's slower, it corrects errors and edits DNA polymerase 3. And also it removes and replaces the RNA primers with DNA base pairs. So it has two different functions, both of them super important to DNA being created. Finally, well, we have two more things to talk about, but the last thing to color, at least, don't you love my coloring skills, is DNA ligase. You know what ligase's job is. You've hopefully, oh no, those are posted after this, but there's the Amoeba Sisters module that I talked about, where they have him as a yellow guy holding a glue, and he's so excited that he's gluing things together. That is obviously a misrepresentation of how he works. But what DNA ligase does is it binds all the fragments together. So it joins, why do I keep closing this? Joins Okazaki, the K is right there on the border, Okazaki fragments of who, the leading or the lagging strand? Okay, of the lagging strand. Good. And it does this by creating pretty much individual phosphodiester bonds, but it joins unattached fragments. Ooh, I wrote that really close together. Joins unattached, there's two words there, fragments by forming missing phosphodiester bonds. Yay. So let me just back off really quick here. All right. Where we see how much we've covered. All the ones that are responsible for the creation of DNA. And then very briefly, I only have one thing to say about exonucleases. They aren't even, unless I just keep missing them. I don't see a number 11 on here. But an exonuclease, it is an enzyme. These are enzymes. Enzymes used as repair mechanisms for DNA polymerase. I'll write it out. 3. So it is an additional repair step for all the errors created by DNA polymerase 3. Okay, and then you should have a very big and beautiful DNA replication form for you to study and also work with while you watch the videos and prepare for your lecture exam 4. I hope that you enjoy this because you're going to do it again for protein synthesis. So thank you. U.